Think for a moment about how we experience the world.
Hmm.
I mean, so much of it really comes down to what we see.
It really does.
Now imagine kind of extending that vision,
Yeah.
pushing beyond what our eyes can naturally do.
Yeah, and that's where optical instruments come in, isn't it? It's a real testament to, well, human ingenuity. This drive to see further or to see things that are incredibly small.
Absolutely. And in this deep dive, that's exactly what we're exploring, some of these, well, frankly, amazing inventions and also the incredible optical instrument we all carry around, the human eye.
Mm-hmm.
We'll start with something simple, the magnifying glass, and then kind of journey through cameras, compound microscopes, telescopes.
And we'll definitely look closely at how our own eyes work and, what happens when vision isn't quite perfect.
We've got some great material here that really shines a light on how these things function.
Yeah, and what's fascinating, I think, is how different they seem on the surface, but, well, they often rely on the very same basic principles of light.
Right. So our mission today is to give you a clear understanding, sort of highlight the surprising connections, the key differences, but without getting, totally lost in the weeds with technical jargon.
Exactly. Keep it clear, keep it focused.
Okay, so let's unpack this, starting with something most of us have probably used, the magnifying glass. Trying to read that tiny print or watching a jeweler look at a gem.
Yeah, or even just looking closely at an insect or a leaf, that's the simple microscope, or magnifying glass.
Its construction seems pretty basic, right? A convex lens, thicker in the middle, usually in a frame, maybe with a handle.
That's pretty much it. The magic, so to speak, is how that convex lens bends light.
Okay, so when you hold it close to something small, you're putting that object within a certain distance, the focal length.
Precisely, within the focal length of that lens.
Right.
And focal length is just the distance where parallel light rays converge after passing through the lens. A shorter focal length means, well, more bending power. And when your object is inside that distance, the lens bends the light. So your eye perceives an image that looks bigger, it's upright, and it seems further away than the actual object.
Ah, okay. So it's a virtual image, not one you could project on a wall.
Exactly. Virtual erect, meaning upright and magnified. Your brain creates that image.
And if you want more magnification, you need a lens that bends light more sharply. So a shorter focal length.
You got it. Shorter focal length, greater magnification.
Okay, that makes sense. Now let's shift gears a bit. What about capturing images permanently? Let's talk cameras.
Right, the camera. A huge leap, really, recording visual information. Fundamentally, it's a light-tight box, usually black inside to stop stray light bouncing around. And it has a convex lens at the front, and then traditionally light-sensitive film at the back. Or, a digital sensor these days.
And there are other bits too, right? A focusing ring and that diaphragm thing with the whole
Yeah.
aperture.
Yes, the aperture, exactly. And the shutter, of course, which controls how long the light hits the film or sensor, plus the viewfinder, so you can actually see what you're pointing at.
All crucial. So the basic idea is the lens focuses light from the object onto the film.
It forms a real inverted, so upside down and diminished or smaller image on that film or sensor. And usually the object is further away than twice the focal length of the lens for this to work well.
Okay, inverted and smaller. And that focusing ring you mentioned, that changes the lens to film distance.
That's right. You adjust it depending on whether the object is near or far, just to get that sharp focus right on the film plane.
Makes sense.
And the diaphragm, with its aperture, works a bit like the iris in your eye. It controls how much light gets through the lens. You need the right amount for a good exposure.
Right. Too much light, it's washed out. Too little, it's too dark.
Exactly. Then, after the shutter does its job, exposing the film, there's that chemical process, developing it to get a negative.
And from the negative, you make the positive print, the actual photograph.
That's the traditional process, yeah. Digital is obviously different, but the optical principles are very similar.
So we've gone from simple magnification to capturing scenes. But what about seeing things that are just incredibly tiny? Way beyond a magnifying glass.
Ah, now you're talking about the compound microscope. That was revolutionary. It opened up a whole hidden world.
Yeah, I mean, think about biology, medicine.
Yeah.
Before microscopes, we had no idea about bacteria, cells.
Exactly. It didn't just let us see tiny things. It fundamentally changed our understanding of life itself. Huge impact.
And they look much more complicated than a magnifying glass. Lots of parts.
They're certainly more complex. You've got two main lenses. The objective lens, that's the one near the specimen or slide,
Yeah.
and the eyepiece lens, the one you look through. They're in the main optical tube. Then you have adjustment knobs, usually a coarse and a fine one for focusing.
Right, I remember those.
And a revolving nose piece often holds several objective lenses of different powers. You've got the stage for the slide, a condenser and diaphragm underneath to control the illumination.
And a light source, or a mirror, right?
Yeah.
Plus the main frame, the arm, and the base.
Precisely. Quite a few components working together.
So how do those two lenses, the objective and the eyepiece, create such high magnification?
It's basically a two-stage process. First, the objective lens, which has a very short focal length, forms a real, inverted, and already enlarged image of the tiny specimen. The specimen itself is placed just a tiny bit beyond the objective's focal point.
Okay, so the objective makes a bigger, real, upside-down image inside the tube.
Correct. Then that image acts as the object for the eyepiece lens.
Oh, okay.
And the eyepiece, which also has a fairly short focal length, basically acts like a simple magnifying glass on that first image and magnifies it again.
So you get double magnification, sort of.
In effect, yes. The final image you see is virtual. It's still inverted relative to the original specimen, but now it's highly, highly magnified.
So the microscopic world appears upside down through the scope, but incredibly detailed. Amazing.
It really is.
Okay, let's swing to the other extreme now, looking at things that are incredibly far away. Telescopes.
Right. Telescopes let us bridge those vast cosmic distances, make distant objects like planets and stars appear nearer, bigger. Think about Galileo.
Yes, seeing Jupiter's moons for the first time must have been mind-blowing.
Absolutely. Changed everything. It showed not everything orbited Earth. Like the microscope, it uses lenses, but they're arranged differently with different properties for a different purpose.
I usually picture a long tube, big lens at the front, smaller one at the back where you look.
Yeah.
And often on a tripod or something.
That's a good picture of a refracting telescope, yeah.
Yeah.
The main parts are the objective lens. That's the big one at the front. It's convex, but with a long focal length. Its job is to gather as much light as possible from that faint, distant object.
Okay, long focal length for the objective.
And then there's the eyepiece lens where you look. That's also convex, but it has a short focal length. It magnifies the image formed by the objective.
So opposite kind of setup to the microscope's objective lens in terms of focal length. How does this pairing work for distant objects?
Well, light rays from a very distant object arrive essentially parallel. The objective lens, with its long focal length, collects these rays and brings them to a focus, forming a real, inverted, and actually highly diminished image right at its focal point.
Diminished, so the first image is tiny?
Very tiny, yes, because the object is so far away, but it's concentrated light. Now, this tiny real image becomes the object for the eyepiece.
Okay.
And since this image is formed at or very near the eyepiece's own focal point, the eyepiece acts like a magnifying glass again, just like in the microscope. It takes that tiny image and creates a final virtual image that's magnified and still inverted.
So the view through a standard telescope is also upside down, like the microscope.
That's right, for a basic astronomical refractor, yes. And while that final image looks much, much bigger than the object does to your naked eye, it's usually still technically smaller than the actual planet or star itself.
Of course, yeah. But it lets us see details we'd never see otherwise. Resolve craters on the moon, rings of Saturn.
Precisely. It's about angular magnification, making things appear larger in our field of view.
It's just incredible how these tools extend our senses, exploring the tiny and the vast. Which brings us naturally to our own built-in optical instrument, the human eye.
Ah, yes, the eye. A marvel of natural engineering, often compared to a camera, and for good reason, but it's so much more complex and adaptable.
It really is. It has that lens system focusing light onto a sensitive screen, the retina, and just packed with intricate parts. You've got the sclera, that tough white outer layer for protection.
And at the very front, the transparent part, the cornea. Also protective, but it's where light first enters and gets bent.
Okay. Then inside that,
You've got the choroid layer just inside the sclera. It's dark, full of blood vessels, provides nutrients, and importantly, absorbs stray light, like the black paint inside a camera. Prevents blurring.
ah, clever. And then the retina itself at the back, the screen.
The light-sensitive screen, exactly. Where the lens focuses the light to form that real, inverted, and diminished image.
Inverted again, just like the camera.
Yep. And the retina has specialized areas like the macula for sharp central vision, and within that, the fovea packed with cone cells for the absolute finest detail and color vision.
You mentioned cones.
Yeah.
There are rods too, right?
Yes, rods and cones. Rods are super sensitive to low light, give us night vision, but mostly black and white. Cones need more light, but they give us our color perception.
Fascinating. And what about the iris, the colored part?
The iris acts like the camera's diaphragm. It's a muscular ring that controls the size of the pupil, that black hole in the center.
Right, the pupil gets smaller in bright light, bigger in dim light.
Exactly. It regulates how much light reaches the retina. Appears black because the inside of the eye is dark, absorbing light.
And behind the iris and pupil sits the lens itself.
The crystalline lens.
Yeah.
It's convex, made of this transparent, sort of jelly-like flexible material. Its main job is to fine-tune the focusing of light onto the retina.
And it's held by ligaments and muscles.
Suspensory ligaments hold it, and they're attached to the ciliary muscles. These muscles are key.
Why are they so important?
Because by contracting or relaxing, the ciliary muscles change the tension on the ligaments, which in turn changes the shape of the flexible lens, making it thicker or thinner.
So that changes its focal length.
Precisely. That's how the eye focuses on objects at different distances. It's called accommodation. It's an incredible ability.
Wow. And there's fluid inside, too.
Yes, two types. Aqueous humor, a watery fluid between the cornea and the lens, helps maintain pressure and shape. And then the vitreous humor, a clear gel that fills the main eyeball behind the lens, keeping it plump and transmitting light to the retina.
So that power of accommodation, changing the lens shape, lets us shift focus from, say, reading a book to looking out the window almost instantly.
Almost instantly, yeah. It's remarkable, but it's not limitless. There's a closest point you can focus clearly without straining, typically around 25 centimeters for young, healthy eyes. The near point, or least distance, of distinct vision.
Okay, so even our amazing eyes have limits. And sometimes things go wrong, leading to common vision defects, like myopia.
Right, myopia or short-sightedness. People with myopia can see things up close quite clearly, but distant objects look blurry.
Why does that happen?
Usually, it's because the eyeball is a bit too long from front to back, or maybe the cornea or lens bends light too much. So the light from distant objects focuses in front of the retina instead of right on it.
Okay. And this is common in younger people, often.
Tends to appear in childhood or adolescence, yes.
And the fix is usually glasses or contacts with concave lenses.
Correct. A concave lens diverges light rays slightly before they enter the eye. This effectively pushes the focal point back, so the image lands sharply on the retina.
Got it. No, the opposite problem. Hypermetropia, long-sightedness.
Yes. With hypermetropia, distant objects are often clear, but it's difficult to focus on nearby things. They appear blurry.
And the cause is the opposite, too. Image focuses behind the retina.
Exactly. The eyeball might be a bit too short, or the lens system isn't powerful enough, doesn't bend light enough, especially for close objects. It's often associated with aging, as the lens naturally becomes less flexible, reducing that power of accommodation.
Makes sense. And the correction here is?
Convex lenses. In glasses or contacts, a convex lens converges light rays before they enter the eye, adding focusing power, helping to bring the image forward onto the retina for near objects.
It's really neat how we can correct these issues with just the right kind of lens.
It is. Basic optics providing elegant solutions.
Looking back, it's quite striking how many parallels there are between cameras and eyes, microscopes and telescopes, and our own vision. They all use lenses to bend light, forming images.
Absolutely. That core principle of using lenses or curved surfaces to manipulate light and form a focused image, usually real, inverted, and diminished in the case of the eye and camera, is fundamental.
But there are key differences too, right?
Oh, definitely. The eye forms a fleeting image on the retina, constantly updating, processed by the brain into our conscious perception. A camera captures a permanent record, either chemically on film or electronically on a sensor.
And the focusing mechanism.
Right. The eye focuses by changing the shape and therefore the focal length of its own lens, that accommodation we talked about. A typical camera focuses by changing the distance between its fixed focal length lens and the film or sensor plane. Different strategies to achieve the same goal: a sharp image.
So just to kind of wrap up the main points.
Yeah.
We look at the magnifying glass, single convex lens, virtual magnified image, the camera convex lens, light type box, capturing a real inverted diminished image permanently.
Yep.
The compound microscope, 2 lenses, objective and eyepiece, giving huge magnification of tiny things, but the view is inverted.
Correct.
The telescope, again, 2 lenses usually, objective and eyepiece, bringing distant objects closer, making them appear larger, also typically inverted view.
Got it.
And the human eye, our natural camera, with its cornea, iris, pupil, adaptable lens, and retina, forming real inverted images with that amazing power of accommodation.
And we touched on the common defects, myopia and hypermetropia, and how diverging concave lenses or converging convex lenses can corrupt them.
That power of accommodation in the eye really stands out as remarkable.
It truly is. The dynamic focusing range is incredible.
Okay, here's something to really think about as we finish up. Consider how these instruments haven't just, extended our sight. They've fundamentally altered our perception of reality itself.
That's a great point. The microscope didn't just show us cells, it led to germ theory, revolutionizing medicine and hygiene.
Right, and the telescope didn't just show us planets. It completely reshaped our understanding of the cosmos and our place within it. It challenged centuries of belief.
So the provocative thought for you, the listener, is what comes next? What future breakthroughs in optics, perhaps seeing in ways beyond visible light, or integrating vision tech directly with our minds, how might those further reshape our perception of reality? What new hidden worlds or cosmic truths might be waiting to be revealed?
That's definitely something to mull over.
Indeed. Well, thank you for joining us on this deep dive into the fascinating world of optical instruments and the human eye.
Yeah, thanks for listening. We hope it shed some light on the topic and maybe encourages you to explore the physics of light and vision even further.